Heard about PCR biosystems?
There's quite a buzz about us.
Hop on over to our website to find out more.
Hello everybody and welcome to Pint of Science 2021. Today's show will cover the fascinating topic on the many ways by which fruit flies have helped us understand science, from biology, physiology and the human body to traveling in space. My name is Stella Kuchiku and I'm a lecturer in biological sciences at Medway School of Pharmacy. So this evening we are coming live to you from the University of Greenwich Medway campus. Before I introduce our speakers, I'd like to encourage you to keep in touch with us via the comments window. Please let us know where you are listening from, and if you have any questions for our speakers, please type those in as we go along. We will have a dedicated question and answer session at the end of the talks. The show this evening is a double act by Dr. Fani Papagiannouli, who is a lecturer and principal investigator of the Fruit Fly Lab at Medway School of Pharmacy, and Ms. Maria Alvarez, a postgraduate researcher in the Fruit Fly Lab. But without further ado, it is my great pleasure to bring to the stage our first speaker for the evening, Dr. Fani Papagiannouli. Hi, Fani. This virtual stage is now all yours.
Thank you very much for the kind introduction. I have to say it's quite remarkable how fruit flies led to so many discoveries over the last 100 years. So I'm really excited to talk to you about Drosophila today and introduce you some of the most important findings that shape biology, genetics, and physiology as we know it today. So Drosophila melanogaster, as the name implies, is a dew lover, and they have a dark gut, which is more prominent in newly hatched flies. They are fruit flies, so we find them on fruit, where they lay their eggs, in the vinegar, but in the wine also as well. So they're invertebrate, they lack the skeleton that we have, and they have 2 wings, and therefore are called dipteran flies. They grow happily in 18 to 25 degrees. Now, despite the striking morphological differences between flies and humans, there are also a number of other obvious differences. For example, the generation time in Drosophila is 10 days, while in humans is 25, and Drosophila has 4 chromosomes, while humans have 23. If we take a closer look at the life cycle of Drosophila, we will see that this goes through 3 distinct stages. First of all, we have the flies that lay the embryos on the food, and these develop over the next day and give rise to first, second, and third instar larvae. So when third instar larvae grew enough in size, they crawl out of the food and they become immobilized and pupariate. After 3 to 4 days, depending on the temperature that they grow, the pupae will hatch and the male or female will come out of the pupal case. Now, medicine and science in general actually have a strong interest in understanding how our bodies function in order to use this knowledge to improve the quality of our lives. For this reason, scientists have used a great number of animal and plant organisms, or otherwise called model organisms, and we in the lab were using Drosophila for our research. One of the questions that people very often ask me when I tell them that I work in Drosophila is to what extent the findings that we have in Drosophila are also relevant to human biology. And I think in order to answer this question, we need to take a closer look at the eukaryotic cell. So the eukaryotic cell with its nucleus has striking similarities between Drosophila and humans. And this is based on their organelles, the membrane that surround the cell, and also on the cellular content. And this is further reflected on the functions of the cells. For example, if we look in our nucleus, the DNA, or the genetic material, is a common alphabet for all living organisms. Moreover, cells divide in a stereotypic pattern, which is called mitosis. Further on, cell connectivity, or otherwise the way that cells stay together within tissues and organs, is also highly conserved. And cell function, meaning also the way that the cells communicate with their environment and import and export molecules inside and outside of the cell, this also follows a very stereotypic pattern, suggesting that the findings that we have in Drosophila can actually be transported to higher organisms, including humans. But what's hiding behind the more than 100 years of research in Drosophila? I think to answer that question, we have to look at two timely related aspects. One is why researchers initiated work in Drosophila at first place, and the second one is why researchers continue work in Drosophila nowadays. So researchers started working in Drosophila to answer simple curiosity questions, and back then, one of the burning questions was how do we inherit the characteristics from our parents, and in some cases, diseases as well. Drosophila was also available in the nature, so it was easy to collect, and it was also easy to keep in the lab. Moreover, scientists were able to find good visible markers for the eyes, for the wing, the hair, or the body color, some of which you can see nicely illustrated in these cartoons, which are also very helpful for our work. Nowadays, Drosophila is still a very popular organ system because it's inexpensive to keep in the lab, it has a short generation time, and we have no ethical restrictions as we have with other organisms, such as, for example, mice and rats. Moreover, we have a great wealth of genetic tools developed through all these years of research, which allow us to study the function of cells, tissues, and organs, and manipulate them also in a very precise way. But what is actually the most important thing, in my opinion, is the fact that Drosophila genes have high homology to many of the human genes, and therefore we can mimic human biological mechanisms in the flies, and thereby model human diseases. Now, if we look at the use of fly from a historical perspective, it was actually Thomas Morgan who first used the flies in his lab, and in 1910, he was able to show that genes are located on chromosomes. You may think that this is a trivial information for nowadays, but in order to appreciate this knowledge, we have to look at that time. And what we knew back then was that Charles Darwin, in 1859, published his work on the origin of species and the importance of natural selection, while Gregory Mendel formulated the fundamental laws of inheritance, which are actually valid until today. However, scientists back then didn't really exactly know what the genes are. Following on the work of Morgan, one of his students, Sturtevant, managed to map the genes on the chromosomes in Drosophila. And this is very important if you keep in mind that in order to cure a disease, we have to know which is the gene that affects it, but also where we can find it on our genome. So Morgan received the Nobel Prize for his work on chromosomes in heredity in 1933, while Müller in 1946 got the Nobel Prize for his work in X-ray radiation and how this can cause mutations. However, at that time, scientists didn't know the exact relation of genes to the DNA. And it was in 1953 when James Watson and Francis Crick, based also on the work from Rosalind Franklin, published their work on the double helix model for the DNA structure. And this elicited the development on very important molecular techniques for sequencing the DNA in 1977 and 1980, but also on amplifying the DNA with a technique which is called PCR. And I'm sure that nowadays, because of the pandemic, the majority of the people now know what the PCR technique is. So this important technology allowed the sequencing of the Drosophila genome in 2000, which was followed in 2003 by the sequencing of the complete human genome. Very important milestones for both Drosophila and humans. So shortly before that, in 1995, Lewis, Wieshaus, and Ullmann Volhard, received the Nobel Prize for finding the genes that regulate the body pattern in the flies and how these are genetically controlled. In our century, we have the Richard Axel, who received the Nobel Prize in 2004 for his work in the olfactory system in Drosophila, Hoffman in 2011 for his work in innate immunity, and Hall, Rosbach, and Jung for their work in circadian rhythms in 2017. However, research in Drosophila, as in many other cases, goes hand in hand in innovations in other fields, and microscopy is certainly one of these. So in 1665, Robert Hooke discovered the first cell, and it was in 1673 that Leeuwenhoek visualized bacteria and protozoa. And since the 1950s, light microscopy is actually routinely used in labs and diagnostic centers to look at cells and tissues. While, on the other hand, the invention of electron microscopy, the confocal laser scanning microscope, and super-resolution microscopy revolutionized the way we look nowadays at cells, cellular events, and how we can investigate the different pathways in the cells. This is further reflected in very important Nobel Prizes, which were awarded to excellent scientists for the innovative microscopes that they found, but also microscopic techniques and ways to visualize components in the cell's life, like, for example, the green fluorescent protein. But what can we actually learn from Drosophila? Or even better, I would say, what have we already learned? So Drosophila is an excellent model system to study aging and longevity. We can uncover the mechanisms that underlie progeria, for example, and aging-related cardiac diseases. We can study circadian rhythms and how the day and night cycles affect our biological clocks and sleep. We can also study the early stages of heart development and cardiac function. Drosophila has made very important contribution in understanding the immune system, inflammatory mechanisms, including wound healing. And a lot of labs nowadays are working on host-parasite interactions. We can also study the muscular system, which is prominent if you think of the ability of the flies to fly and the need that they have for coordinated movements. Further on, in the last decades, we've learned a lot on the nervous system by studying Drosophila, for example, by modeling diseases such as Alzheimer, Parkinson, and epilepsy. And we can also use flies to study learning, memory, including drug addiction. The Drosophila flies have a gut which has striking similarities with the human one, and therefore we can use the flies to study the microbiome, nutrition, and how all these affect physiology and other metabolic diseases such as, for example, diabetes or even obesity. A very important advantage of using Drosophila in research is also the fact that by using Drosophila, we can replace, reduce, or refine, in some cases, the use of laboratory animals, such as, for example, mice and rats. Drosophila can also develop cancer, so they can have metastatic tumors. And here in the pictures on the left side, you can see wing primordia. So these are little organs that give rise to the wings of the adult flies. And next to it, you can see a wing disc in which has developed cancer. So you see this amorphic overgrowth mass of cells that represent the tumor. Drosophila flies also have stem cells, and this is very similar to the humans, where adult stem cells reside in mature organs and tissues, where they can replenish cells after injury, after aging, or in, for example, tissues with a high turnover, such as, for example, the gut. But I will discuss a little bit more about these two aspects close to the end of my talk. Drosophila is in recent years actually intensively used for drug screening. So when we want to test, for example, new therapeutic reagents for their toxicity, we can use the Drosophila flies, and then we can assay also other parameters like, for example, the lifespan, the well-being, the viability, and in some later cases, it's actually used also for personalized medicine to cure cancer. NASA established a very interesting program to test the effect on the flies on microgravity, extreme conditions, and low Earth orbit. So they actually want to find out how this affects the physiology and different biological parameters in the flies. Finally, we've learned a lot using flies on how our body plans are established, and such a prominent example are actually the Hox genes, for which I will discuss a little bit more right now. So the Hox genes are very conserved across the animal kingdom, and color-coded, you can see here the Hox genes and the structures that they give rise in the flies. So you can see that the Hox genes are in more than one cluster in humans, and they specify morphological structures along the anterior-posterior axis of the organism. And this is the reason why Hox genes are actually called master regulators. Now, any kind of defects that affect the function of the genes lead to visible defects. And here you have an example of a Hox gene that causes sympolydactyly if you look on the left side of the panel. And if you look on the right side of the panel, you can also see extra digits. Now, in the case of the flies, Hox genes lead to transformations. And here you can have an example on the top of a wild-type fly with a little structure which is very small, and it's called haltear, which is now transformed to a second pair of wings, which explains very well why these genes are actually called master regulators. So right now, I'm going to hand over to Maria, who's going to tell you a little bit more about how we keep the flies happy in the lab. Hi, Maria.
Hi, Fani. Thank you very much. Indeed, I'm going to be talking about everything that entails keeping the flies in our lab. And as you can see from these pictures, we keep our flies in two big temperature-controlled incubators. And we normally keep them at exactly 20 degrees. However, this can be changed easily as needed. Within the incubators, we have the flies organized in boxes, with flies with the same purpose in the same box, so that it is easy for us to know roughly where everything is. And within these boxes, we keep at least 2 vials of each fly stock to ensure that they are as safe as possible, and should one of the vials get contaminated or die out, we have the other one to rescue the flies. Now, the environmental conditions in which flies are kept play a really big role in their survival. The standard conditions to raise flies in the lab is at 22 degrees with 60% humidity. And with these conditions, the full life cycle of a fly from an egg to an adult fly will take around 12 days. This, however, can be changed to our needs, as their developmental speed is greatly dependent on temperature. So if you wanted the flies to grow more slowly, you can lower the temperature to, say, 18 degrees. And here it will take at least 20 days for the full life cycle to occur. On the other hand, if you increase the temperature to, say, 25 degrees, the opposite will occur and the life cycle will be shorter, around 9 or 10 days. This can be really handy if you need flies to grow quickly for a certain experiment. However, it does also have its drawbacks, such as infections that are easier for them to grow, and even parasites such as mites to appear. So it is always really important to keep a close eye on your stocks and make sure that they are doing well in between food changes, or how we like to call them, fly flipping. And you're probably wondering, what is fly flipping? Well, fly flipping is a crucial part of every fly lab, and it entails transferring flies from one food vial to a fresh one in order to maintain their growth rate. This is usually done every 4 weeks when we are just trying to maintain the stock as it is. However, if you're trying to amplify a stock, you can flip the flies as often as every other day. This is because even though it might take a while for the new flies to hatch, leaving the flies in the fresh vial for a couple of days is enough for them to lay eggs that will eventually lead to new flies. So even though it might take around 2 weeks, by the end of it, you can have multiple vials of the fly stock of your choice. And on this video here, I demonstrate how the fly flipping takes place, although it is pretty straightforward. All it entails is gently tapping the vial with the flies in order to get them to come away from the cotton, and then, as the name indicates, just flipping them onto a new food vial before quickly closing it with a cotton ball, as seen here. And you might be wondering, what is in these food vials? Well, the most common basic fly food will contain inactivated yeast, yellow cornmeal, some form of sugar, such as sucrose, and agar to keep it all together. However, with regards to how we obtain the fly food, there are different routes that you can take. You can make and pour the food yourself using the ingredients required, which is normally done when you need to create fly food with an additional specific ingredient that is not common in traditional fly food, or you can just buy the pre-made food. Now, here you can see a box which usually comes with 100 food vials ready to use. And this is how all the individual vials look like before putting the flies in. And on this picture, we can see how much of the food they actually eat. On the left is a vial that was flipped over a month ago, and on the right is a recently flipped one. And you can see that these flies really do enjoy their food. Now, you're probably wondering how we handle the flies, because unsurprisingly, they have wings, and they can very easily fly away if you're not careful. So when we need to handle the flies, we just anesthetize them. Thankfully, there are several methods that have been developed to do this, each with its own pros and cons. One of the most common ways to do this is by cooling them down by placing them in a freezer until they are asleep, and then keeping them on ice whilst handling them. Another very common way to do this is by using ether, although this is slightly toxic. However, what we use in our lab is carbon dioxide. This works really well at keeping the flies immobile for longer periods of time, although it can be more expensive to do, as you will need special CO2 blocks and, of course, a CO2 source, which is usually a tank, which needs to be secured by clamps and safety checked regularly and all of that. But if you are using CO2, it is pretty straightforward. All you have to do is open the valve and ensure that the right amount of CO2 is coming out, which should be just a gentle breeze on your hand. And then you just stick the nozzle end of the plastic tube into the fly vial, ensuring that the cotton is always at the bottom. And the reason why we do this is so that when the flies do fall asleep, they fall to the cotton and not to the food where they can stick. It also always helps to gently shake them so that they come away from the walls. Once they are asleep in the vial, you just pour them onto the CO2 block and you're good to go and work on them for as long as you please. Now, from this video, I'm sure you can see that it really doesn't take long for the flies to fall to the cotton, and they're able to stay like that for long periods of time, making it really easy for us to see them under the microscope and carry out any experiment that we need. So now that you know the basics about fly maintenance, I thought I would tell you some random fun facts that you might not know about these fascinating little insects. The most interesting one, in my opinion, and the main reason why we use flies in research, is that around 60% of the fly genome is homologous to that of humans. And even more importantly, that around 75% of disease-causing genes in humans actually have functional homologs in the fly, which means they're very good to study. Another fun fact is that females can actually lay up to 100 eggs per day in the right conditions. And even virgin females can lay eggs, although they obviously do not produce any flies as they're not fertilized. Also, funnily enough, the fruit fly does not actually eat fruit. It eats the fungus that grows in the rotting parts of the fruits, although it does lay its eggs inside of the fruit, and this is to ensure that eventually the larvae have something to feed on when they need it. And maybe flies aren't that different to humans after all. I mean, even male flies like a bit of alcohol when they've been rejected by a female. A study at the University of California actually found that when given the choice between food soaked in alcohol and its non-alcoholic equivalent, the decision made will be greatly impacted by whether it has mated recently or it has been rejected by a female. And the ones that have been rejected by a female choose the alcohol-soaked one, probably because the consumption of alcohol activates a reward pathway in their brain. So it basically makes them feel better. Although, unfortunately, humans don't have wings, which obviously flies do, and they get full use of them. It has even been shown that some flies can beat their wings up to 220 times per second. But anyway, enough about fly fun facts and more about actual flies and how they look. So here are some pictures to compare and differentiate male from female flies. As you can see, the females are generally longer and males are darker in color. Males also have a more rounded end, which is also darker in color to that of the females, and this is what we use to tell them apart under the microscope, as it is the most distinguishable feature. Now, this is quite weird, but it is also really important to be able to distinguish flies which are virgin from those that are not. This is important specifically when setting up new crosses, as it is the only way to ensure that the progeny is exactly what we want. Thankfully, it is not too hard to do, as both male and female virgin flies have a black spot in their abdomen, which is meconium, circled in black. This is only present in recently hatched and virgin flies and is usually not there for very long, so there's a very small window of time to collect them from when they hatch. Now, when the flies first hatch, because they have been inside of the pupal case, their wings are folded together as seen on the picture on the left. It usually doesn't take too long for the wings to expand and take their normal shape, but this is also another easy way to tell the recently hatched flies apart from the older ones. However, sometimes we need to be able to tell flies apart regardless of gender, and this is why there have been so many markers that have been found. And you might be wondering, what do I mean by markers? Well, markers are just specific changes in the fly's DNA, known as mutations, that give rise to certain characteristic, which is known as a phenotype, and this helps us distinguish them more easily. This is also really important for us because they are frequently used to trace otherwise invisible DNA mutations that we might make. So for example, I might make a mutation in a gene, but this doesn't necessarily change the appearance of the fly itself. So often we join both mutations together, so we know that the flies that have the different characteristic are also the flies that have the mutation that we're actually interested in. And they can get very creative with these mutations. I mean, there's one that's called J-Lo. Anyway, here we have some of the most common markers used in wings. Let's start with serrate. In this mutation, the wings have multiple notches, with the most prominent one being on the inside bit of the wing, and some smaller one towards the outside bit of the wings. This mutation, although it is really easy to spot, does have a lot of variability, so the flies that do have this mutation will not necessarily look the same. And the same happens with vestigial, where the wings and the halters are deteriorated and it looks like they have teeny tiny wings. Curly, as the name indicates, means that the wings are curled upwards and outwards slightly. And interestingly, this is affected by temperature. The higher the temperature that they're in, the curlier that the wings get. And finally, wingless, which, again, as the name indicates, means that one or both wings are missing and instead transform into thoracic tissue. So they end up having an enlarged thorax, which appears to have a double hump when you look at it from the side. But enough about wings, let's look at some eye color markers. Now, just like with wing markers, eye color markers are simply mutations that change the color of the fly's eyes. Let's start with brown, which again, they were very original with the names, means they have light brown eyes that interestingly darken with age. Although when they are crossed with certain other colors, such as scarlet, they can actually result in white eyes. Now, sepia has very dark brown, almost black eyes that again also darken with age. So at first, they are more of a dark brown and turn basically black within a few days. On the other hand, scarlet changes very little with age. It might darken slightly, but it is normally indistinguishable. And as you can see, flies with this mutation have bright red eyes that are very easily distinguishable from others, especially from wild-type flies. And finally, we have white eyes, which, as you probably guessed, means the eyes are white. Although in some cases, the mutation can actually show a slightly orange/yellow eye color, depending on the mutation and the background itself. Lastly, I'm going to look at some body markers. As you might see from this slide, flies have two main mutated colors, which are ebony and yellow. Ebony has a much darker pigmentation than that of the wild type, which runs throughout most of the body. And in this marker, when the flies first hatch, they already look darker than normal anyway, but as time goes on, they get darker and darker, including their wings. On the other hand, flies with the yellow marker have a much paler appearance than that of the wild type and look like their body is yellowish. Now, markers do not need to be in adult flies necessarily. Larvae can also have markers too, such as tubby. With tubby, larvae, pupae, and even adults are shorter and stout. However, this is most obvious when they are at the larvae stage, as seen in the picture, and they get progressively less obvious as they go through the life cycle. It can actually be quite hard to distinguish them once they're adults, so it is always best to separate them at the larvae or even pupae stage to ensure that you're getting correct ones. And finally, with black cells markers, we see that they display dark speckles on the larvae and to a lesser extent in the pupae and adult too, although it is again most seen at the larvae stage. Interestingly though, this does change with temperature again, and flies raised at an elevated temperature will have fewer speckles than those at a lower temperature. And with that, I would like to welcome back Fani for the second part of our talk, where she will be talking about what we have learned in the lab using Drosophila.
Hi, Maria. Thanks a lot for the nice presentation. So for the very last part of my talk today and today's presentation, I will show you a little bit the kind of questions that we ask in the lab using Drosophila. So in the lab, we work with Drosophila in all different stages. So we use the embryo, we use the third instar larvae, and we also use the adults. For example, in the Drosophila embryo, we can study how the first cells are formed. For example, here in the diagram, you can see a monolayer of cells, and below you can see a close-up of such an embryo with a nuclei, this ellipsoid shape stained with fluorescent green, and the membranes of those cells shown here with fluorescent red. So at an early stage, before the cells are fully formed, we get a picture like the one in the bottom, where you see the nuclei now in blue. And then you can see on top of those nuclei sitting these cytoplasmic structures we call microtubules, and these are the ones that sort of guide the formation and the growth of the membranes. And the membranes then grow in between the nuclei and then close the cells. And here on the bottom in red, you can see the membrane front, which will lead to the formation of the cells. We can also study third instar larvae, and we can use this stage to study cancer. So here you can see third instar larvae. On top you can see a normal one, and below you can see larvae with an enlarged body shape. And this is because these larvae have tumors which affect their internal organs. And we can actually dissect these organs and look at them under the microscope. So if you look here, for example, you can see the brain and wing primordia from a normal fly, while below you can see the brain and wing primordia from third instar larvae that have developed tumors. And you can see how bigger these organs are and all the overgrowths that they have. And we can still use this tissue and ask several questions and study them further. However, the large part of the things that we're doing in the lab is related to the germ cells. So the germ cells in all sexual reproducing organisms are very important because these are the cells that will produce the sperm in the males and the eggs, or otherwise called the oocytes, in the females. Therefore, the germ cells are very important for reproduction because these are the cells that give rise to our progenies. So in the Drosophila embryo, the germ cells are set aside at the posterior part of the embryo, while the rest of the cells, which we call somatic cells, will give rise to the body. And if we look at the germ cells under the microscope, here you can see a close-up of the picture with the germ cells stained with a green fluorescent protein, while the somatic cells are shown in red. So at the end of embryogenesis, the germ cells coalesce together with some of the somatic cells and start building the gonads. The gonads continue to develop through the instar, larvae, and pupal stages. And then when the fly hatches, in the case of the males, we have a fully formed testis, which you can see here in the diagram. So these testes are localizing in the abdomen of the flies. And if we look at them under the microscope, here you can see the germ cells now showing in purple color, while the somatic cells, which are these thin indentations between the germ cells, they're shown here in green. So what we actually want to understand in the lab is the communication and the equilibrium that it's set up between the germ cells and the somatic cells. And one of the questions that we ask is what happens when this communication is disrupted. So one case scenario is that the signal is totally gone. In this particular case, the germ cells don't know how they can produce the sperm and sort of over-proliferate, but they stay small and undifferentiated. The result, of course, is that these flies cannot produce sperm and they're infertile. On the other hand, we can also have the situation where the signal is too strong, and then the germ cells realize that something is going wrong and they actually die. And you can see here that in this particular case, on the right side, we have less germ cells or less of the purple cells. And then in this case, again, the sperm is not produced and the flies are infertile. So this indicates us how important cell communication is in order to have homeostasis in the tissue, fertile animals, and a healthy reproductive system. And this is very important because all the things that we're studying in the flies refer to very conserved pathways and cellular events. And therefore, most of the things that we learn in the flies, we can actually transport and test later on in higher organisms, including humans. And by that, I would like to wrap up today's presentation by thanking all my academic teachers and mentors during my undergraduate studies, during my PhD, and later as a researcher at the University of Heidelberg, Stanford University, and the University of Cologne. Some of my former colleagues have, in the course of the years, become very good friends as well. So this work led to very important publications, together with the support of some of the funding agencies. My special thanks will actually go to my PhD supervisor, Bernard Mechler, and my former PI at Stanford University, Margaret Fuller, as these were the ones that supported and shaped my research the most. So now you can find us at Medway School of Pharmacy, the Universities of Greenwich and Kent. This is where I started my own lab in September 2019. Maria, who gave you the nice presentation about how we work with the flies in the lab, is actually a PhD student who joined the lab in September 2020, in the middle of the pandemic. So she is actually a very brave student. And by that, I would like to thank you very much for your attention, and we are ready with Maria to take questions.
Thank you both for the fascinating and very informative presentations. We have received quite a few questions from our audience, but if I may, I'd like to kick off the Q&A session with a general question, probably for you, Fani. In the first part of your talk, you showed us the plethora of research areas the fruit flies have been involved in. However, I was wondering if there are any limitations to Drosophila research, and if so, what are they?
Thanks, Stella, very much for this question. Actually, if we want to be fair, we shouldn't only talk about the advantages, but we should talk about the limitations. So I think in Drosophila, it's very difficult, or let's say impossible, to model processes which have evolved from the vertebrate lineage. And this, for example, includes work in the mammary glands, how calcified bones are set up, or some of the spinal malformations. Now, if I can give an example, heart, for example, is a different situation, because we can model the very early stages of the cells that specify the heart up to the formation of the tube, which is very similar to what the Drosophila has. On the other hand, when we want to talk about the 4 chambers of the heart and all the later developmental stages, this we cannot model in the flies. So there are also some other examples. For example, when we talk about functions that require this complexity that humans has and Drosophila doesn't have, then again, we have some limitations. And there are also some examples where other model organisms, like, for example, yeast or C. elegans, may be more appropriate. For example, we study cell division, scientists study cell division in yeast routinely, I would say. So it has always, every model system has its pros and cons. Yeah.
Thank you. We have a question from E.L. Priest, I think. So they're asking, are there any everyday challenges when working with Drosophila, and how do the logistics work? I suppose the second part of the question is answered by Maria's talk, but can you name a single, maybe big challenge that you face in the lab every day?
I think for a person that starts in the lab, and it's quite a young researcher, I think handling the flies and learning how to keep them happy in the lab is a big challenge. I think for more mature scientists, I think the challenge is always to find the answer for the questions you're looking for. And it happened to me many times that I have some sort of sleepless nights where there is a bothering question, or a pathway or something that we want to find out, and we're desperately looking to find a solution for that. So this is actually the challenge, yes. And once, of course, we have our story ready, we have to convince the editorial boards, right? Because these are the ones who will publish our work. And at the very end, we have to convince the funding agencies who fund our research.
Thank you. I think the next question is very timely. Kate is asking, is Drosophila helping in COVID-19 research and in the fighting against the pandemic?
Yes. The short answer is yes. Of course, Drosophila, as I already mentioned, doesn't have the complexity that we have in humans, but we can study individual aspects of COVID-related functions. For example, scientists have expressed proteins of SARS-CoV-2 in flies. They're studying their function, and then they were testing which, for example, drugs can cure those diseases. And this is a way to answer these kind of questions relatively quickly and cost-effective.
Thank you. Next question, it comes from Laura, and I think it's directed to Maria. She thanks you for the great talk, and then she asks, what happens to the flies if the temperature goes above or below the range of 18 to 25 degrees Celsius?
That's actually a very good question. Thank you very much. Well, if you go a little bit outside of that range, there really shouldn't be too many side effects other than a change in the growth rate, which I already mentioned. But for example, if we go above, if we go to temperatures of around like 35 degrees or so, the flies will survive still, but you will start to see some side effects such as sterility. And above that, it really depends on the type of fly, but usually they don't survive easily above 40 degrees. And if we go on the other side of the spectrum with lower temperatures, again, with like 10 degrees or so, they will survive, but they will move a lot less to conserve energy and heat and all of that. And as mentioned, if you go below like 0, it will actually anesthetize them, as I mentioned. So they'll just be fully asleep. And if they spend too much time asleep, they won't actually recover from that. So it depends how much it varies.
Thank you. Another actually quite interesting question from Eleanor. How is NASA using the flies for space research?
This is actually very interesting. Thank you for the question. So what NASA really wants to find out is whether they can predict if when they send astronauts like long-term in microgravity conditions or in extreme conditions like radiation, right? What would be the effect in their bodies? And this is the reason why they use Drosophila to, let's say, learn from the flies and then transfer these and be more prepared. So what they actually do is that they have prepared a set of devices for bringing the flies and feeding them in the space, a little bit different than what Maria showed today. And then they look at different physiological parameters. For example, what about the well-being, the life cycle? Do they age earlier? Do they get any mutations on their DNA or the expression of the proteins? How does their immune system works? Can they fight, for example, infections easier or not? Do they get even any changes in behavior? Because we can also study behavior in Drosophila. How fit are they, the flies? Is their metabolism affected? Do they eat more or less? So all these kind of natural habits that would affect the life quality of an astronaut, these are the things that they also check in the flies as a way to prepare themselves for what will come ahead.
Very interesting. One more question I think we have time for. Rofi3 is asking, how are the Drosophila specific body markers created in the first place? And there is a second question to that. And how frequently can they be found naturally in the wild?
That's also a very good question. Actually, most body markers are just random mutations that happen naturally, and someone found them and just put a name to them. So usually the name given to it is actually related to the phenotype seen when the DNA is mutated. So for example, with white, which I spoke about earlier about an eye color, it's actually called white because the mutation causes the eyes to be white. And this was actually discovered by Thomas Hunt in 1910, and he denoted it as capital W, which is what we still actually use to the day when talking about this mutation in the lab. And another example is with vestigial, which again was also found by Thomas Hunt and Calvin Bridges in 1919. And they were actually the ones to name it like that. And with regards to how often it occurs in nature, there's not really one fixed number that you can give because it depends on the mutation itself. Each mutation will have different probabilities. But obviously there will be a higher probability of getting this mutation if the parent has the mutation, then the progeny is most likely to have it if it's dominant and whatnot. So there's not a fixed number. It depends.
Okay, thank you. I think I will take on from you mentioning the white eyes, and I will ask Simon Scott's question. Are white eyes only seen in males?
No, they're seen in both males and female flies. Yeah.
Stella, if I may say something here, I think Drosophilists have a sort of a weird habit. They give their flies funny names. So that's why sometimes you have homologous genes between Drosophila and humans, and of course scientists in humans and other organs have a serious name, but Drosophilists have usually funny names. So one such example, for example, is what Maria mentioned before, Jerry Law. So that actually means Jennifer Lopez, because the wings are quite enlarged. And we also have genes that are called Ken and Barbie. So based on the phenotype that the flies have, they actually like to recapitulate this. And coming a little bit to Simon's comment on that, actually what the white gene does is that under normal situations, like the wild type form, it creates red eyes. It's just that when it's mutated, it causes the white. So this is, again, another sort of weird way of Drosophila people naming their genes by their phenotype of the mutation.
Thank you. We have, I'm going through the chat now. We have a question I saw from Andrew Tipman. He's asking, how can parasites gain access to these effectively isolated fly populations?
I think, well, one of the parasites that the Drosophila have in the nature, but we also can get them sometimes infected in the lab, are the mites. These are special mites, but they're not much different from the ones that we get in our beds and pillows, actually, in our everyday life. In principle, they can't go through the cotton balls, but this requires on our side to be also very careful when we handle them in the lab. For example, we should flip the flies relatively frequently. We shouldn't let the vials get too old because then the mites have an advantage over the flies. And we have to keep everything very clean because keep in mind that they can't go through the cotton, but we take the flies in and out of the vials. So we use them under the stereo microscope, we dissect them, we do things with them. So we have to keep a very good cleaning condition in the lab and avoid also other insects to come in the lab. And when we receive flies from other labs, we actually keep them in quarantine and make sure that they are clean and ready to go in the fly lab.
Thank you. So I have two questions. They are similar, actually. They're from Obed and E.L. Priest. They're asking, what are the regulations regarding Drosophila? So the ethics of, using Drosophila in the lab.
Drosophila do not have any ethical restrictions. They're not vertebrates. And as insects, they don't fall in the regulations that we have with any other laboratory animals, such as mice, rats. Yeah. We are lucky in that respect. And one has to say that when we use them, they actually don't suffer, right? Whatever experiments we do, they're sleeping with the CO2. And even when they dissect them, they're also sleeping. So
they're
when
anesthetized fully.
yeah.
Yes. Thank you both. I think regarding questions, I think this brings us to the end of our tonight's show on Pint of Science 2021. At this point, I would like to thank again our speakers, and I would like to thank you, the audience, for tuning in this evening and making this show even more interesting with your brilliant questions. And until next year, I think it's bye from us at Medway School of Pharmacy. Take good care of yourselves and see you soon. Bye for now.
Take care, everyone.
Thank you.
Thank you, Stella, as well.